I want to start by thanking James and Stephanie for agreeing to do this presentation today so early in the year and so early in the morning in California. So we try to set this up so we could span at least California to Izmir, to Turkey, in reasonable time frame. And we have some, Emily's even on from Hawaii at 6 a.m. But this is the time that seems to work best. So this is our special interest group by the CASP organization on ensembles of conformations or collections of conformations. And as I was saying to James, as we thought about where the future for CASP challenges are, considering the great success in basic modeling that we saw over the last few years, we started to think about, could we actually model energy landscapes, distributions of protein conformations? And it's very challenging. And one of the real challenges is how do you assess such predictions? And let's see here. Yeah, so today we want to talk about a problem which is a little bit of a thorny problem, which is really how do you represent the structures when you have collections of structures? And you might think it's pretty straightforward, but we really don't have standards in for example the protein data bank, or as we share information in this project. So I'm actually going to give a short talk today about a recent perspective that we wrote about the challenges here, and then James and Stephanie have come up with some potential solutions to some of these problems. So I want to talk about this with the idea that we might adopt some of these methods in the CASP 16 activity on multiple conformations. And I'll say we're recording this, and comments in the chat are welcome, and the PowerPoints and the video will be available. You should actually join this email list here, which is pretty specific to this CASP SIG, even though it has Montelione Lab as the name. And then we'll let you know when the YouTubes are available. You could even search for the YouTubes. And we also have a LinkedIn group where we share information in this general topic. So the organizing committee put together this kind of vision statement, which I'll read: recognizing that biomolecules do not have static structures, but rather adopt a distribution of conformational states. The CASP SIG aims to evaluate if a critical assessment of predictions of conformational ensembles or multiple conformational states is feasible. Is there a way we can do for multiple conformations what has been done so successfully for, say, static structures? The SIG will consider developments in the fields of structural and computational biology as they evolve over the next few years, and try to develop protocols for assessing the accuracy of multiple or ensemble model predictions. And what we're talking about here is the idea that you might have two conformations or two states, like the classical adenylate kinase crystal structures, or perhaps tens or hundreds of conformational states that might describe the properties of intrinsically disordered proteins, linkers, or intrinsically disordered regions of proteins. So we have a steering committee made up of people who have quite a bit of expertise and interest in this area, and I want to thank these people for their time. And we meet about every quarter and just see how this is going. We're working under the general auspices of the CASP project. So John and Christoph and Andre play a key role. And as I'll explain, we're going to be doing a CASP exercise where Andre and Christoph will be playing a significant role as we evolve this. And I just want to highlight that there are actually 3 special interest groups organized by CASP. This one focused particularly on alternative conformational states. Sometimes it relates to AI methods, sometimes it relates to biophysical or experimental methods, or even molecular dynamics simulations. Then there's a second CASP specific to AI methods in structural biology, how they relate to CASP predictions, but also how it relates to protein design And so I believe the next meeting is next Wednesday, Philip Kim, Machine Learning Methods for De Novo and Peptide Design. And Arnie Ellison, who's on the meeting here, has been organizing that. And we also have a CASP RNA special interest group on modeling RNA and nucleic acids generally. And Rachel Kretsch and others organize this. And in fact, I think they've been doing it every 2 weeks. I haven't been going to most of these, just occasionally, but I want to highlight that also because very important activity. So in CASP 15, we did have a activity on conformational distributions, and we had 3 or 4 targets, and we made an effort to assess these and got some experience with it. And this is the published paper, which if you're interested, you could have a look at. And now what we want to do is to now move this into a CASP 16 activity. So CASP 16 is this year, and the season of CASP will be about May to August. And the idea here is to put out targets for modeling predictions and assess the targets. Well, the typical assessment is against, the hidden X-ray crystal structure or cryo-EM or NMR structure. And here it's a little trickier because, why do we believe the way the experimentalist modeled? Why do we believe the way experimentalists modeled the data? Why is that the ground truth? And so this is really one of the thorny issues in this activity, and it may actually doom the activity, like how do you really assess against ground truth? And we've had some discussion, and what we've decided is we have to really assess against the data, somehow taking the models and back calculating the data. And that means that we need to collect data for targets that are the blind targets, and then release the sequences, and then assess against the data. So the data providers, therefore, will have to play a key role, both in telling the community what kind of data they're going to be assessed against, and what they should be trying to model, and also in helping my group do the assessment, because we're not experts in all these techniques and all these assessment methods. So we have to access some cryo-EM data or NMR data. In any case, that's what we're going to try to do this year. And then eventually the data will be released on the CASP prediction site so people can then do their own assessments against the data. And I hope this will really drive the field in a good way. So we need to collect targets and data, and I actually have 2 groups who I've already spoken to. One of them will be some cryo-EM, multiple conformational data. Another one will actually have to do with disordered linkers between domains, where people have generated or will be generating data about relative positions of domains with respect to one another, determined by the flexible linker. And I haven't confirm the dates, but these people will present in this venue, and it'll be recorded so that predictors can understand what is the problem, what's the domain, and what is it that how are their predictions going to be assessed? And for the data providers, I want to say we've had some great experience with this. By providing data to the CASP community, you develop very interesting results with your data and learn new methods, and often it's the opportunity to develop new collaborations. So I really have found this to be very rewarding, and I hope we'll get some good data sets for this. I don't expect we'll get more than half a dozen targets in doing this, but we'll see how it goes. So that was kind of my short introduction. It went a little longer than I wanted it, but I think the next part will go a little faster, and we'll definitely have at least 30 or 35 minutes for James's talk. So usually we trying to have 1 presentation per meeting. I decided in this one, I wanted to talk a little bit about our own, at least a perspective that we've done, a little bit of our own work, and then go
Guy,
to,
your screen share stopped.
no, it's just a blank screen. Good,
Oh, okay, gotcha.
thank you. Yeah, so this is the perspective. This is the graphical abstract from the perspective we published in recently in Current Opinion in Structural Biology, and it is just that, an opinion. I want to describe some of this, and I want to start with this idea of a single conformation. Oops. A single conformational model. That's smaller. Do you see that bar there? I try not to see. Maybe you don't see it. It's good.
See a bar on the left.
Yeah, we do.
I don't know how to close that.
But you know, mostly that just tells us you like to use Firefox. So we're okay with it.
Okay.
Oh, it disappeared.
Okay, good. So single conformational states of biomolecules. And, you know, we have dealt with this issue, particularly in the NMR community, but anytime you model ensembles, you generate a set of structures, a set of models, but that aren't supposed to represent alternative states or multiple conformation. They're really just meant to represent the uncertainty in the model. And I've had a very hard time conveying this idea to the broader community, particularly with NMR structures. But AlphaFold came along, and one thing that they did was provide this metric of uncertainty of residue or atomic positions, the PLDDT score. So the PLDDT score is the probability of a local distance difference test scores. The local difference distance test is really comparing the distance matrices between two models. And if they're exactly the same, so it doesn't require superimposition. If the distance matrices are exactly the same, the LDDT would be 100 or 1.0, and they're very different, it would be 0. And one thing that CASP introduced in the prediction activities was not only should you predict the structure, but you should predict the reliability of your structure in different regions. And the wonderful thing that was done by DeepMind in training AlphaFold was not only to train it to predict the coordinates, but to predict the uncertainty in the coordinate positions. So we have this model confidence score, at the probability of having a LDDT or a accurate structure. And now we're quite familiar with this. We look at the structure, if it's a low score, and we might color it yellow or orange, we know we better be careful with that part of the model prediction. We're very comfortable already in a year or two with this PLDDT mapping. So for NMR structures, and it's not only true for NMR, but whenever you're generating models of a protein and you generate it multiple times to get an estimate of the uncertainty, we'll often then deposit in the PDB an ensemble, a set of structures. But this set of structures is not meant to represent the real conformational diversity of the structure. It's really meant to represent, like the PLDDT, the reliability of the structure. Here, the structure is very well converged, and we say, well, the data seems to, from the data, I have a good confidence in that modeling. And this part is not very well converged. And all it really tells me is that it's just not well converged. I just don't know exactly what the structure is. These are not meant to represent the different states of the protein, as some people have incorrectly interpreted. It does actually relate to flexibility, but it's not in any way, no effort is made here to model different conformational states such that some mix of them fit the data. And yet, despite the similarity of these two ideas, this is very easily conveyed. This one, people still struggle with. They take the first model, or they don't even use the NMR structures because they don't know what to make of this uncertainty measure. It would be simple enough to just map this uncertainty onto a representative structure, but it's rarely done. And I'll just point out, this is an example of comparing some some NMR-derived models with AlphaFold-derived models for the CDK2 associated protein by some graduate students and Teresa Ramelow in my group. And the point here is that in the upper left, you see the NMR structure, and the not well-defined regions in yellow are indicated. And I'm still having trouble with this bar. Whereas in the AlphaFold 2 structure, again, the regions with low PLDDT score, it turned out to be, there's a very good correlation between the poorly defined, probably dynamic regions and the low PLDT score. And there's been a number of studies, and we've done some, showing very strong correlations with low PLDDT and real dynamics. So these are the points here. The atomic variance in a single conformational model is highly correlated with the locations of structural flexibility in the structure. Very often this variability is due to real dynamics, but it's not in any way a quantitative measure of the dynamics, and it could even be due to just incompleteness of the data. But these conformations in these regions of the model are not usually meant to represent the actual conformations that are in dynamic and equilibrium. In fact, the structures in these regions are really just inaccurately defined and sometimes not even physically reasonable structures. And so for that reason, when we do structure quality assessment, we generally exclude those not well-defined regions from the quality assessment metrics, such as packing scores or Ramachandran scores. So to generate accurate models of these conformations actually requires some kind of multiple conformational state modeling. So this leads to a big problem. Number one, while the structural biology community has quickly adopted the PLDDT metric as a measure of local model reliability, people remain confused about using the experimental single-state models deposited, for example, by NMR and other biophysical studies. So the uncertainties in the modeling the atomic positions are not reported in a standardized format. And we tell our undergrads, please report your error bars. Take this back, minus points. You don't have your error bars on your chart, but we're not showing our error bars. In fact, it is a challenge to convert the data into real statistically reliable uncertainties, and that remains a challenge. But at least we have some measure of uncertainty by these well-defined and not well-defined regions. So this led us to this, see if I can close this. No. I'm just having a little bit of problem here, sorry. So we have this pipeline for annotating atomic variance in single state models. First, we have to have conventions for saying what regions are well-defined and what regions are not well-defined. And in the NMR community, we spent a lot of time on this, and we've developed standards. These are some of the programs, and if you use these different programs, I won't get into them, you get pretty similar results that you can by convention say, this variation is well-defined and this is not well-defined. And once we define that, and these are actually superimposition independent methods, so that once we define that, we can then superpose the structures using only the well-defined regions and identify the one conformation most similar to the others. We call that the medoid structure. That's the representative structure that's most like the others in the well-defined regions. And then using that medoid structure, we can calculate variance, atomic variance, atom by atom, and represent that graphically. So this is the medoid structure with the uncertainty represented graphically in color and in thickness. And because this is an atom-specific measurement, we have the information not only per residue, but per atom. So this is a very valuable way to represent these ensembles. And we developed, we have a general software tool called PDBstat which does a lot of different things, but one thing it does is it carries through this pipeline. You take the PDB coordinate file in MMSIF or legacy format, and it does this process to calculate the medoid conformation, the atomic variance, and then defines well-defined or not well-defined residues or atoms using these conventions, which I say are quite similar to one another. So this is a way to address that problem. But here I illustrate another protein, another study. And in this is actually the dengue virus protease. And what you see here is actually what the modeling has done is not generated a single state, but tried to fit the data to multiple states. And so here we have NOE data of this drug, a large drug bound to the protease, and the NOEs that are generated are inconsistent with one another. And we're able to parse those into consistent groups and solve two structures from the same sample. So now we're not talking about uncertainty per se, but really two sets of structures, each with their own atomic uncertainty. So this is what we mean by multiple conformational states, as distinguished from the single conformational state with uncertainty, as in the previous slide. And these are actually, and I'm not going to get into the details of the data, but they're actually experimental methods which we can use to measure the rates of interconversion between states, and here we're assuming two states in the modeling, and also get the populations and the rates. These are nuclear relaxation experiments, Carpacell, Milbom-Gould, CPMG experiments, by which we can actually characterize rates of interconversion, Milbom-Gill. And we get populations and we get rates of exchange. So now what we want to do is to represent these data in our atomic coordinates that we deposit in the PDB. So this is actually really a challenge that we're facing. So these multiple conformational states, they're described in the literature as alternative conformations, switched folds, metamorphic states, chameleonic states, conformational ensembles, conformational excited states. These are all kind of the same idea. The various methods for interpreting experimental data in terms of multiple states models are evolving. And in this CASP SIG, we're trying to explore those methods and their connection with prediction and modeling. So there's a current pressing need to standardize the representation of such multiple state models and their corresponding data in structural databases and across the structural biology community. So this actually begs the question, what is a conformational state? And I think in statistical mechanics, there probably are more rigorous definitions than we're using here. And it is a little bit of a fuzzy term in some ways. So conformational states are distinct conformational models or collections of models that in principle can be distinguished by some experiment. Structural models can be characterized as single state, like I was talking about earlier, or multiple state, but it depends on the nature of the data or how you interpret the data. Multiple state models may constitute, say, a pair of conformations. Adenyl A kinase was an example, or pairs of conformational collections, or collections of many conformations, say, disordered polymer chains. And as I understand it, the distinction between states and even the counting of the states is determined by the interpretation used in actually modeling the data. And I'll show you what I mean. So this is some kind of one-dimensional projection of an energy landscape. And you could see there would be some low energy conformations that would be populated based on Boltzmann. And I might do a study and modeling where I get three states, one down here, this one a little less populated, and then the third state, like adenyl A kinase. Or maybe I do have different kind of data and I have different kind of interpretation, and I can actually model each of these individual minima. So instead of 3 states, this is a 12-state fitting. Or maybe I have a lower resolution. I'm only fitting it to 2 states. So the actual number of states is not something that you determine, let's say, by looking at the coordinates in the end. It really depends on how the scientist has interpreted the data and what kind of experiments have been done in generating that. And I'm just about done here. So in NMR, we do have sometimes a separation of states. If we have slow interconversion between 2 environments, let's say the methyl cis to the oxygen and the methyl trans to the oxygen, we'll see 2 separate resonances. But as you interconvert more rapidly, they broaden, they coalesce, and then they'll come to some population average weighted frequency. So, of course, here you might say, I can interpret that as 2 states. But even in this regime of intermediate or even fast exchange, we may be able to pull out information about the different states contributing, like I was showing for the dengue virus. So again, there are certain spectroscopic features that we can immediately call 2 states, but even when we see one set of peaks, we may interpret it as 2 or 3 or multiple states. And so this is being done, and there is more and more literature. And here are some examples from the PDB of multiple state models, multiple conformations determined within the same sample, and often, not always, but often with assigned populations, like I was showing from those CPMG experiments or other methods. This top one, 7UBG and 7UZL, that's a cis-trans isomerization that distinguishes the states. Whereas the upper right, that's the dengue virus, that's fast exchange, but we still could characterize 2 states. And one of the problems here is we don't have a standard for representing these kind of information in the PDB. In some cases, these are represented by 2 separate PDB IDs, like the top left. In other cases, like the lower left, we define them by chains, 1 PDB ID, but each chain represents the different conformational states. And then this one, 7R95, there are 2 states, but they were just dumped together in the PDB file without a clear designation about what the different states were. So we lack a way to share and to support such data. So big problem number 2, multiple state models are not represented in the PDB in a standardized way, at least for NMR-based models and other such modeling. And then for each of these models of the multiple state, we also need to report the uncertainties, and that's not done in a standardized format. We have an alt-log tag, and perhaps James and Stephanie will describe some ideas of that, but we don't really have a good way to manage that. And so in this review, we talked about how we might handle such data and the importance of capturing both the raw experimental data as well as interpreted data, as well as the models that are associated with the states. And I refer you to the article if you want to learn more about these thoughts. But fundamentally, it's not a solved problem. And so James reached out to me and said that he had some thoughts about how to address this problem. And he and Stephanie have explored that and put out a preprint, which I think is very interesting. And it might be some ideas we want to adopt in the SIG or more broadly across the community. So James, I apologize, I went a little longer than I planned, but please pick it up from here.
Great. Maybe if you can release the screen sharing. There we go. You know, most of what we're going to talk about today is going to be done by Stephanie, a former student and scientist in the lab, really touching on similar themes to what Guy has just introduced. I'd say, you know, our perspective is really shaped very much from the experimentalist end, and as well from rather than sort of an NMR dominant view, where I would say most of the time we're in a relatively data poor sort of data to parameters ratio for modeling atomistic structures. Our experience has been mostly from X-ray crystallography and EM, where I would say the data is actually sort of dramatically overdetermined. And I'd say the problem is usually in representing multiple states, not just where things blur out, which we've discussed extensively, but much more so on the end where the data are in fact too rich to be explained by one state. And Stephanie will explain a little bit more about what that means. I'll also just introduce the idea that we really are looking as we go sort of from left to right on this slide here. We're looking at a conformational ensemble that exists inside a crystal that through the sort of power of X-ray crystallography or EM, undergoes this transformation in crystallography. This is literally a Fourier transform in terms of a diffraction pattern that generates what is sort of the raw data. And we have that data measured with very high precision. But then when we compare a structure to that data after the inverse Fourier transform, creating a density map or comparing it directly to the integrated intensities, the error in our structure is much larger, generally, 5 to 20 times higher than the precision of the measurements themselves. And so this creates an interesting thing where ground truth for things like AlphaFold is actually already wrong compared to the experimental data. And when we compare an AlphaFold prediction or any other method, generally the quality of the precision between that prediction and a structure, even for well predicted structures, is an even higher error than that. So the question that I think this community can start to address, and this is relevant for the single structure prediction community, but I think even more relevant for the ensemble or alternative conformation prediction community, is how can we start moving from predictions not through a avatar of ground truth, which is the PDB structure, but start to think about comparing much more to the raw data, either the integrated intensities, the density maps, or the diffraction patterns or particle stacks themselves. And so as we start to do that, I think that there are some really interesting representation problems that need to be solved on how these ensembles or structures are represented and encoded. And that's what Stephanie has been doing really exciting work, a lot of hard thinking to figure out, and that she's going to talk about today. So I will hand the reins over to Stephanie Wankowicz, and she will present for the remainder, and then we'll answer some questions together.
All right, thank you so much. All right, thank you all for joining. Happy New Year. So today, as Jamie kind of really set up, we're going to be talking about ideas that we have on improving modeling, but also encoding of conformational ensembles. And this is really coming from the realm of X-ray and cryo-EM. And I really think that, what is preventing us to kind of getting to the structural ensemble that we know existed richly in our experimental data and translating this into predictive models is both, is a dual problem. And we've made a lot of progress, both work from us and many other labs on the modeling side. And there's a lot of momentum in that. But I think for us to actually take advantage of a lot of that modeling, we need to think about how we're actually encoding this in the experimental data, experimental models that we have. And then how can we also think about how the experimental models are going to be connected to these predictive models. And so I want to start out by, just starting at the basics of, the fact that structural data is capturing many conformations, the conformational states that experimental that proteins sit in. And here I'm showing a video of a cryo dragon, and this is from Cryo-EM, of showing sort of large conformational states that proteins exist in. And while this is, very relevant, we sort of think about the conformational states that proteins have on a much more atomistic scale. So thinking about sort of the range of different times and length scales that conformational heterogeneity exists on, this is incredibly important. And as the experimental techniques become more advanced, I think thinking about how we connect sort of these different length scales of conformations are really important. And so not only thinking about sort of these large states that proteins take on, but these very small different states that side chains and backbones take on. And when we think about these smaller conformational states, we think about it in terms of the ability for X-ray crystallography and cryo-EM to actually capture a lot of these ensembles. So within X-ray crystallography or cryo-EM, and within a single crystal form, or within a single classification of in cryo-EM, side chains can exist in multiple different states. And these different states, even different rotamer changes, often does not disrupt the crystal lattice. It does not interpret us from classifying cryo-EM into multiple different classifications, but it's critically important in the enumerations of states that proteins have. And importantly, in X-ray crystallography and cryo-EM, we're not restricted by the time scale either because of the way that we're collecting the data. And so we're able to capture much more of the ensembles of the different states that the side chain sits in. But, I think for anyone who has downloaded a PDB structure of X-ray crystallography or cryo-EM, you very rarely see this. You very rarely see side chains modeled into multiple conformations, and this is really just because of the way that data is encoded. It's incredibly messy. It's not that modelers are lazy in any sense, but it's very difficult to do this by eye, by identifying these multiple conformations that side chains sit in, especially when they occur very infrequently or at very low occupancy across the data. But if we think about sort of, what static modeling is doing compared to what the protein ensemble that exists within the structures, the static modeling does not adequately capture this. And so if we think about modeling a side chain into a single conformation, it's looking at the averaged conformation that the side chain sits in. This is generally sort of your highest signal compared to noise. And so this is why this is modeled, but it's really leaving out a lot of the rich data. And so what we really are kind of bread and butter is taking these static structures and turning them into what we call multi-conformer models. And so multi-conformer models are modeling in side chains or backbones into multiple conformations as supported by the experimental data. And so thinking about how the different coordinates as well as the occupancy of each side chain or each backbone atom fits into multiple different states. And by that, we're explaining a lot more of the conformational ensemble that exists within the experimental data. And so I just want to take a moment and kind of contrast the multi-conformer models to ensemble models. And I think this is really important thinking about how we're encoding this. So if we think about an ensemble model coming from X-ray crystallography, oftentimes you have many copies of the protein, and this can be very difficult to interpret exactly how does a single model fit into the electron density, how does it relate to the different models, and it's very difficult to manually manipulate. On the other side is sort of our multi-conformer models, where we try to be a lot more parsimonious in how we're explaining the conformational ensemble. But this allows experimentalists to be able to manipulate the model much easier as they would wish. And it also is just a much simpler way, but still explaining much of this conformational ensemble that exists. And so, as I said, most of X-ray crystallography and cryo-EM are not modeled in any sort of ensemble or multi-conformer modeling. And this is mainly because of the difficulty of being able to detect this. And so what we have done over the past about 10 years is develop a software tool that allows you to automatically model in a multi-conformer structure from sort of your single static structure. So we don't take from experimental data and create these multi-conformer structures, but if you're able, if the experimentalist is able to create a baseline model, we then can use some really powerful optimization algorithms to look in a lot more detail into these experimental data and be able to model in side chains and backbones into multiple conformations. So I'm just going to briefly take you through this as I'm talking. It's a freely open source software available on GitHub. And so what we do is, like I said, we take in a model, we break it down into different backbone, different residues. And first we do some backbone sampling. And then we get all of the different conformations of the backbone, and we use an optimization algorithm that where we ask, based off of all the conformations of the backbone that we just sampled, how best do these combinations of the backbone fit with the experimental data that we have? And then we choose backbones, and then we do a very similar thing for side chains, where again, we're sampling extensively, and then we're asking the optimization algorithm, how best do all of these samples of the side chain best fit into the experimental data? And importantly, with the way that we're encoding the conformational heterogeneity in the PDB and is carried over into the MMSIF right now, we then have to label it. So we label the different conformations into A or B or C, et cetera, conformations. And then from there, what we do is that we put the protein back together. So each of that is done on an individual residue basis. And the first thing is really looking at making sure that the backbone is all strung together, looking at multiple conformations in the backbone, asking again, which, using our optimization algorithm to identify which best explains the experimental data. And then finally, we use a simulating annealing to determine appropriate labeling. So looking again at 3D clashes or conformations that are going to be touching and asking, how do we label this appropriately? And so this gives us out a full multi-conformer ensemble model. And so looking at some high resolution X-ray crystallography data, our question was, we know that proteins exist in these ensembles. If we're modeling more of this ensemble, are we explaining the experimental data better? And so we turn to this metric called R-free, which is used in X-ray crystallography to ask, what is the fit between our model and our experimental data? And we can see that that QFIT explains the experimental data in about 75% of the structures better than the deposited mainly single conformer structure. So anything below this red line indicates a QFIT multi-conformer model is explaining experimental data better. And so I think this is just, a really important point to, pause on and say that, the modeling that we're doing is really starting to get these ensemble or multi-conformer models are really starting to get at explaining the experimental data better. And that, with continued advancements of this, I think that we will continue to, drop this even more. But we're running into a lot of issues of exactly how do we interpret this and encode this into PDB or an MMSIF format. And so beyond explaining the experimental data, I'm just going to show you some examples of, the kind of hidden alternative conformations that we can find using QFIT. So the mesh here is your electron density. Any green spots indicates that our model is not explaining the experimental data well. Red spots indicates that our model is over-explaining the experimental data. And by using QFIT, we reduce a lot of these red and green spots, and we're identifying, some very obvious alternative conformations of the protein, but we're also finding a lot more subtle conformations. And when we think about relating the protein ensemble back to protein function and interpretation, even these very subtle conformations are incredibly important. And so beyond improving the model, we improve the geometry of the model. And so right here, I have plotted the bond angles and bond lengths, with green being the deposited and purple being the QFIT models. And you can see that the QFIT models kind of left shift this, identifying that they're closer to ideal bond lengths and angles. And across for those who are familiar with the mole probity score, which is looking at the overall geometry of structures, we're improving this altogether. And so if we think about sort of why this is occurring, if the proteins that we're collecting in the experimental data are existing in multiple conformations, but we're trying to shove multiple conformations of a side chain in, by explaining it using one conformation, we're often going to strain these bond angles and lengths. And so what QFIT can do is really nicely fix this by fitting in multiple conformations of side chains that are still are accurately explaining this experimental data, but are producing much more physically relevant models. And we can also do this in Cryo-EM, although this is, kind of at the beginning of this, both due to the resolution limitations of the method and being able to identify alternative conformations. But we're excited to kind of push the resolution limits on this side. And so, when we think about the fact that of conformational heterogeneity and thinking about the ensemble that proteins exist on, this is very common, but it's unmodeled in most structures. And so if we look across X-ray crystallography structures across the PDB contains almost all static single conformational structures. So on the X, you have the number of alt locks that exist in a single structure. And then on the Y-axis, the percentage. And, this is a slightly skewed interpretation of this, but almost all structures only have less than 3 alternative conformations in here. But we know that for the most part that, proteins exist in these multiple conformations. So, we can use tools, like QFIT to be able to model this in, but to be able to encode it is also a critical issue. And I think when we think about sort of the encoding, we want to come back to sort of this wide range of conformational heterogeneity that can exist in structures. So not just thinking about sort of conformations on a sidechain scale, but how does this then relate to larger conformational structures? And this is going to become more and more important experimentally. And I think also thinking about this prediction-wise, how does conformational heterogeneity that we're able to capture on this much larger scale relate to conformational heterogeneity that occurs on this atomistic scale? And so if we think about sort of how do we encode this currently in the legacy PDB format, and importantly, this has also been transferred directly over into the current PDB-X MM-SIF format, you basically have 2 options. So you have an alt lock option here on the left, and you have this occupancy option on the right, and these are related. And so you basically can encode an alternative conformation by identifying its alternative locator ID. And this is really the only way that you can communicate to someone who's looking at your model that a side chain or a backbone or a portion of your model is existing in multiple conformations. But we can think about the fact that conformational heterogeneity exists in many different ways. So we can think about this very simple model of conformation A or conformation B, or sort of loop out and loop in. But we can also think about the fact that a lot of conformational heterogeneity is encoded in sort of hierarchical formats. So you can think about a loop being out or in, but then within a single loop region, maybe a sidechain is then in multiple conformations. And so right now, with our encoding of only using A, B, C, D, there's no way to encode any of this hierarchical conformational heterogeneity. And so what we are really proposing is that to amend an MM-SIF to encode this conformational state ID, which can both be have multiple layers. So it can be explain this hierarchical conformation, and is a lot more flexible than the alt lock ID. And so thinking about sort of this in graphical format, you can think about this occurring again on an atom level basis. So the atom the compositional state, which we'll go to in a minute, and then sort of having multiple layers of conformational heterogeneity, explaining a loop out and then a loop in, and again, the occupancy of each state sort of encoded in there. And then thinking about moving on to the fact that, I talked a lot about conformational heterogeneity, but compositional heterogeneity is a huge of growing interest. And so here I'm showing an interpretation where in the experimental data that you have here on the left, you have a ligand, or you can think about this of a subunit of a protein that's only existing in some of the copies of the protein that we're getting our experimental data of, but then this is, averaged over to get the experimental data, and we really need to tease out what exists in sort of the apo or unbound, or maybe only a core part of the protein and it doesn't have this additional subunit in cryo-EM versus sort of this bound conformation. And so again, thinking about how we encode compositional heterogeneity in the legacy PDB format, we again have this alt lock ID and we have the occupancy, but if you're like seeing double, it is because this compositional heterogeneity is encoded in the exact same way as the conformational heterogeneity. And so this becomes incredibly messy. How do we encode sort of compositional heterogeneity, and how is it incorporated with the conformational heterogeneity? Just using sort of alt locks and occupancy is really not enough for us to explain this. And so we really think about this of splitting this out into having a compositional state and a conformational state. And so thinking about this graphically, you can think about having, again, your conformational state of whether this is unknown or bound or unbound compared to the conformational state. And when we think about sort of the heterogeneity that exists together in structures, we can think about this graphically in interpreting both the compositional state of bound or unbound, and then how this relates to a conformational state. And we think that this really explains a lot more of what exists in the experimental data, and that we're really able to push towards in modeling better, but that's really not captured accurately in the PDB format. And by capturing this in the compositional state versus the conformational state, and also having conformational states being hierarchical, we can capture and encode much more intelligently how this is related, and have someone take your model and be able to really interpret this and connect this to prediction model. And so, just thinking about sort of, of interest in connecting this back to protein function, we really need to think about this conformational distribution and how this contributes entropically to protein function, which is really an interest of mine. And so if we think about sort of the ways that we have the ability to do this as a structural biology community, we really need to think about how these structural conformational ensembles can connect this thermodynamics of proteins to structures. And to get there, we really need to be thinking about this entire hierarchy of the conformational states and how this exists in the compositional states. And so thinking about, how we get there and connect to the ensemble prediction, the first thing is obviously improving modeling, being able to interpret a lot more of this underlying experimental data. And as I said, I think there's a lot of momentum getting there, but I don't think that we're really going to get there unless we improve the encoding of these ensembles, and what we're really trying to push for now, and thinking about how we can connect this again to this protein prediction space. And so with that, I'd like to thank the lab. The QFIT code is here, and I also put the title of the perspective piece that Jeannie and I wrote, that goes into the sort of encoding in a lot more detail than I presented here, and I'll unshare and happy to take questions.
Okay, so I think that was great. Thank you, Jamie, and sorry, Stephanie and James. I think people can just ask questions, but if you want to raise your hand, I'll see that and call on you. But we have some time let's go for 10 minutes. We'll go 5 minutes over here with people need to leave, that's fine.
We're
Yeah.
happy to stick around all day. We love talking about this.
I actually will start, which is that, Stephanie, you talked about like in QFIT, that you're now going to sample some different conformations and find the set of those that, fit best to the data. I guess it would be ideal to do that sampling with some sophisticated modeling. So my question is, can you imagine a way to have a CASP type experiment where you have data, maybe we release the static structure or just the sequence, and ask people to generate distributions where they're basically sampling those states you described, and then use QFIT to assess that modeling. Does that make sense, or is that kind of an overdetermined some way problem?
Yeah, let me see if I can state back what I think the question's asking. So basically taking from a static X-ray structure, are there ways that we can predict which residues exist in a multi-conformer state versus not?
Yeah, and I'll say a little different.
Okay.
Is there a way to use the CASP community to explore methods for doing that? That's really my question.
Yeah, so I mean, I don't think I'm gonna sit here and, I do think QFIT is sort of at the forefront of being able to model on this atomistic scale. But I do think that, I'll be the first to admit that we don't get everything that exists in the experimental data. So thinking about sort of waters or ligands or loop structures. And so I do think that the way to go towards this is much more on an experimental scale. I think that we need to create improved metrics to be looking at sort of an experimental compared to a model, whether it's a predictive model or a experimentally derived model. And that's where I would really see the pushing forward, rather than, again, I think that, we could see a lot of improvement by having the CASP community thinking about other ways that they could be solving this problem than we have been approaching. And I think you could make more advances forward that way.
Thanks.
So maybe I'll just chime in with a brief. I think there are two ways to go about this. I agree with Stephanie. The second way, which I'll advocate, I think is the more complete way. But the first intermediate would be finding ways to compare sort of ensemble versus ensemble, or multi-conformer versus multi-conformer at the coordinate and occupancy level. I think that runs into the same problems that we have with single structure issues. And the second is comparing to a raw form of the data and thinking about overfitting as well within that. And, I'd like to have, I agree with Stephanie, let's have our eyes on the longer term prize, but even the first one is hard to implement.
Thank you. So Susan, I saw you raised your hand.
Yes. I apologize. You probably mentioned this, Stephanie, but in your alternative conformers, are you including ones that are in non-preferred but allowed rotamers?
Yes. So our sampling takes, the idealized rotamer state for every residue, and we look at about 50-degree range from that idealized, so about 25 on each side. And then we really allow the optimization algorithm to choose. And we do refinement afterwards. And so there is some correction, but we do see some non-idealized rotamers in our structures. They tend to be about on the same level as deposited structures.
So the reason why I mention it is James and I were having a conversation in the chat. I have two crystal structures. One is 1.1 angstrom, one is 2.8 angstroms of the same family protein. and the rotamer is a 90-degree from the preferred. And there's a tyrosine and a aliphatic chain, and then there's it's an as-pis pair. So it's being forced into that conformer. And of course, AlphaFold 2 gets it wrong, and it's like one of the highest conserved residues in the active site. And so it seems like, I think that these really extreme outliers do exist. And I have no way to like figure out how do you do that in an optimal computational way. I mean, obviously I have experimental data, but what are your thoughts on that?
Yeah, so this is something we've, gone back and forth on a lot of, how widely do we sample and what do we allow, and then allowing sort of the scoring to take place. We used to do 360 sampling of rotamers. And we found that for the most part, there was a lot of mismodeled rotamers. However, the experimental data, like the R-free, tended to be slightly just, ever so slightly reduced in that way. And so, but I don't know what kind of the right answer is. I do think that, one of the things that the advantages of QFIT over an ensemble model is that, we may not capture every single conformation, especially sort of these main outliers, but you can then take it into COOT. And we hope that, by improving that model to data fit more, that some of those other rare, or outlier alternative conformations are going to pop up a lot more, and that you're able to then add it to the model. But for the most part, we're really looking for, just based off of, I don't know if there's like a way to say, without fitting to noise or fitting to, some other spurious things right now. How can we pick up on sort of that real signal?
But I would argue that a true outlier will be rock solid in the density.
Yeah. No, I mean, right now, right, we would just not be even sampling it and even providing it. And, we have thought about ways of how can we use, Bayesian information criteria or other metrics to sort of decide on what is the best map to model fit. And again, trying to get at this like parsimonious model. And that might be something that, we do a sample a little wider initially and kind of narrow in, and we could think about that in that aspect.
Thank you.
I think Keith was next, and then I'm glad Brenda raised her hand because I wanted Brenda to comment on this. Go ahead, Keith.
Yeah, thank you, Stephanie and James, for the great talk. My question is, do you find the alternative conformations that you identify with QFIT, do you find that they're correlated like residue to residue, like through space, and like what ways does protein packing or side chain packing affect the alternative conformations that a QFIT finds? Because my understanding from the talk was that you're optimizing ball and stick models to fit densities on a residue per residue basis, but then some of those alternative contacts are mediated through packing. It's very similar to Susan's comment and example that there's a forced state because of mediated through packing. So is there like new areas to improve QFIT by looking at how residues can be correlated with each other and how their alternative states are they're not on like a per residue basis, they're through packing?
Yeah. So, we break it down to residue states, I'd say mainly for computational efficiency, but that they're, and biologically, right, we do see, relationships between, the ways that proteins are packed are going to differentiate on, where alternative conformations sit. It's not anything that our software takes into account currently, but I think that it's something we could think about, and thinking about sort of moving into these sort of, correlated loop movements that are associated with conformational heterogeneity. It's something that we're thinking about moving forward, but we don't currently do.
So Brenda, also, I'm curious what the view is from, you're still working with the PDB, I believe, right? And
Yeah.
what your view is from the PDB about this representation of multiple conformational states.
Right. That was part of the reason I raised my hand. So with regard to integrative structures, we expanded PDB XMM SIF to handle almost everything that was discussed here today, multiple models in multiple states, models with in multiple states that could have heterogeneous composition, and also conformational ensembles that actually depict the diversity or uncertainty, not exactly in multiple states. So these are the kind of definitions that we had to grapple with over the last few years while we expanded the description, the data definitions for integrated structures. And although that's currently separate from the main PDB XMM SIF, they are all in the same language and can be considered together. And I'm more interested in making sure that we don't come up with some divergent set of definitions here, make sure it's consistent. So anything that will be useful for this group in making sure that the definitions are standard and compliant with the PDB, I'm happy to help with.
It's quite relevant for me because in the SIG, in this CASP exercise, I want to adopt some standard for describing the multiple conformational states that predictors may generate that would then be validated against data. So we need to understand where PDB is at with that and how it relates to some of the ideas James and Stephanie put forward. So is that something that's, I'm sure there's a white paper of some kind. Is that a published paper, or it's how could we disseminate that to this community?
So yeah, so we have a Git repository with the extension dictionary. We have documentation. We do have papers. Another one from the community is currently in process. It will be submitted this coming month. Because ultimately we are going to merge everything. All the integrative structures are going to be part of the PDB. So preparing for that, we're submitting this paper. Again, the extension was created for integrated structures and may not be fully sufficient, but we are happy to create whatever is needed to accommodate the requirements here. For example, I mean, one thing that I saw in Stephanie's talk is expanding the atom site table for metadata about conformational states. I don't know if that is ideal because that's something that pertains to the model. Not sure it needs to, it's at the atom level. So there's a kind of things that maybe can be handled differently, but we probably have to see if the current set of definitions will be sufficient or there's something new that's needed.
Yes, if you could provide a link for that, I would send it out to the email for this group who would be very interested in that, if that's publicly accessible at this point. Right,
sure. Yeah, the Git repository is public. The original paper where we came up with the definitions, that's public. Yeah, I will send all the links.
Maybe I can just chime in that, this is a really urgent thing to solve, not in PDB dev, but in real PDB, and to work really closely with experimentalists, because the revolution in sort of high throughput crystallography means that the majority of structures or data sets that are being deposited in PDB are coming from high throughput ligand soaking experiments. Those almost universally have partial occupancy in terms of composition at the various binding sites, and heterogeneity of conformation as well. And this is probably the most interesting and rich data source for improving potentials for small molecule protein interactions. And right now, it's just a complete clusterfuck to try to data mine that because the compositional and conformational heterogeneity are interlinked in the way we encode it, if they're even encoded at all. So, what we need to do, right, is like something that I think is, attack on multiple fronts, which is one, like change, the standards and integrate these ideas from PDB dev and what we're advocating here. But I think more urgently is we need to make sure that the software that folks are using to refine these structures and to build these models. So basically, 99% of this is encompassed by RefMac, Phoenix, and Buster, and most of the modeling is done in COOT for what is basically the majority of deposit X-ray structures and EM structures, with this ligand conformational and compositional heterogeneity. And so we need to really make it easy for those software packages to adopt these new standards, and so that folks who are depositing these structures can use it in a really facile way. And there's sort of this chicken and the egg thing where when we talk to the folks, developing that software, they're like, we need a way to actually, the PDB to lead. And then, we talk to the PDB and it's like, oh, well, we don't want to complicate things for the software folks. And so I think the real key to what we've written and what we discussed today is something that is sort of backwards compatible with the legacy format, enables sort of facile manual modification in COOT, and has a roadmap to play really nicely and simply with the software packages, in particular in the implementation of this hierarchical representation with something called grouped occupancy refinement, which is implemented in Phoenix, RefMac, and Buster, as well as something that's a holdover from even more legacy software like Shell Excel.
Right. I mean, I see the urgency in the requirement. I'll bring it up with folks here, and I understand what the software people are saying, that it has to be done fundamentally at this data standards level, because only then can the software follow what's the standard. They can't just, I mean, if each one starts doing something, then it's not a standard anymore.
Yeah.
So,
but I think that and look, Andre Shawley's office is, just that way. One of my best friends. I love the guy, but we need to not have this try to solve every problem all at once, because right now we have the potential to solve a very concrete problem in terms of compositional and conformational encoding in crystallography. That is the majority of the experimental information that is going into the PDB. So we can't delay getting this encoding done in a way that enables, I think, what is one of the most important questions, small molecule protein interactions, because we're trying to solve every single problem in integrative structural biology.
no, we don't have to do that, do everything. We can merge in bits and pieces. We can take what we want and not do the whole merging. Depends on I guess, the push from the community.
Yeah. So I'm glad we're on the same page, because we want to push.
Okay, I will bring it up with Stephen and see how quickly we could do that. But I would also like to know I mean, you said there is a definite set of requirements here, which is urgent. Is there some kind of documentation, or what exactly is needed, so I can maybe identify the subset that needs to go in very quickly and kind of push for that?
And this is exactly what Stephanie has written.
Yeah, so I'm happy to send along kind of our perspective piece and sort of toy examples that we've created of MMs.
Yeah, toy examples are the best place to start. So I can look at that and then see what's needed to go forward.
Okay. Yeah,
Please.
I will send that along.
I promised people we would try to keep this to 1 hour, and I went a little over, but it was actually a really great discussion. It was very valuable to have. So this video will be available for people to review, and I hope various people, and I'll be in touch also, Brenda, about related issues. So thank you very much, Stephanie. Thank you, James, and thank you, everyone. Happy New Year.
Thank you.
So awesome. Thanks for catalyzing this community.